for time purposes. So I want to get things started by just saying welcome to everyone. My name is Rachel Robinson. I'm the Chief Operating Officer of MassMedic. I'm a veteran of our webinars, but I haven't had the chance to actually run one for quite a while. So I'm so thrilled to be here with you yet again. It takes me back to, 2020 times when we were all doing webinars. But we're so thrilled to have all of you join us for what is going to be an outstanding program. And I'm so thrilled, like I said, to be a part of it. So just wanted to kind of start off by saying thank you and welcome to everyone. We do have some quick housekeeping notes. If you would like to ask a question during the session, it is highly encouraged. Please use the Q&A tab at the bottom of your screen. That is the best place to pose questions. And then our panelists will get to them either during the presentation or at the end during Q&A, whichever kind of flows with the discussion best. We do want this to be incredibly interactive, so please feel free to pop those questions in, and we hope to hear from you. This is our last webinar of the year, and I'm so thrilled to kind of close things out with a bang. And just to say, again, thank you to Triple Ring for putting this wonderful program together. I know you're going to learn a lot about sterilization, so I'm going to go ahead and turn things over to Sheila to get us started.
Thank you very much, Rachel. Hello, everybody. Welcome to today's MassMedic webinar. I'm Sheila Hamami. I'm the Senior Director for Growth Initiatives at Triple Ring Technologies. And today's topic is entitled Design for Sterilization Enables Accelerated Time to Market. This webinar has been organized today by Triple Ring Technologies and by Dose Insight, both of whom you will learn about in greater detail from our panelists. Triple Ring brings together scientists, engineers, designers, and strategists to tackle the hard technical challenges faced by medtech and life science technology companies today. We have a nearly 20-year history of accelerating early-stage deep technologies off the bench and toward commercialization. And Dose Insight's origin story is out of our own research and development in this space. With that brief introduction, I'm going to turn the conversation over to Daniel Badali, who will be moderating today's discussion. And if you have any questions about how Triple Ring Technologies can help you with your own early stage technology, please feel free to reach out either today in the chat or through our webpage contact us link. So with that, Daniel, to you.
Great. Thank you for the introduction, Sheila. So we're very excited to have some industry leading experts in the sterilization field to participate in a panel discussion about virtual dose mapping. And because virtual dose mapping is a new technology that sort of, it's had a long history, but it's emerging as a commercial technology that's now available to medical device development companies. We thought to start with a brief overview of what virtual dose mapping is and what it can offer you by incorporating it into your medical development process. So with that, I'd like to offer Tobias the floor here to start with a little brief overview of virtual dose mapping.
Yeah, thanks, Daniel. Hello, everybody. See if I get my screen shared. Okay. Yeah, I'm from Triple Ring Technologies. I'm the VP of Technology Development, and I want to give you a quick introduction to Triple Ring. We at Triple Ring, we help other companies to develop products, and we have a deep technology development background. We are located in Newark and Boston with about 100 people, and we focus on life sciences and medical device development. And we also do internal R&D, which is partially funded by government grants, and some of these ideas we think have legs, and then we try to spin out these ideas as independent products. And Dose Insight is one of those. And at Dose Insight, we have developed a virtual dose mapping tool, which I will talk to you about today. And we offer that to medical device developers and sterilization vendors. And you can learn more at our website here what we are up to. Today, I want to focus on design for sterilization and how virtual dose mapping can enable an accelerated time to market for your products. I just, as a brief overview, FDA guidance for manufacturers of sterile devices. And by no means will I give here a comprehensive overview of that, but I try to give some nuggets that are important. For guidance, the manufacturer has to identify the sterilization method that is going to use, basically the sterilization assurance level, basically that is the likelihood that something fails, and then the method of sterilization validation. And then the FDA defines these established sterilization methods that are easier to go through the process than things that are not established. And as established count ethylene oxide, which is a poisonous gas that the product is exposed to, and that takes about 50% of the sterilization market. Then there's dry and wet heat, which is rarely used in the devices we are talking about. And then the other 50% is radiation. And so we are focusing here mostly on radiation sterilization. And radiation sterilization is governed by these two ISO standards, and they're very comprehensive. And again, by no means I go in more details, and there are other guidance documents. So there's a lot out here. But in a nutshell, as a manufacturer, you have to define the verification dose of your device. That means the dose that is required to make your device sterile. So that also means it's the minimum dose. And you also have to define a maximum dose. And the reason for that is that the device or the materials can be damaged if you go over a certain dose threshold. The important thing to keep in mind, it is not enough to just assure that one device can be sterilized. You have to ensure that the whole package of devices that you put in the beam is sterilized. So that is important to keep in mind. So for radiation sterilization, we go to a facility that provides a high energy radiation source that can be either gamma rays, with cobalt-60, or X-rays, or electrons. And here we focus on electrons today. And this intense beam basically irradiates your device, and it will deliver dose to the device. So how do we know that our device received the right dose? And what we really need to do is we need to optimize the dose uniformity ratio. As I mentioned, you can't exceed the maximum dose, and you have to be above the minimum dose. The ratio of the two is the dose uniformity ratio. And the question obviously is, what is optimal? So, I mean, ideally, you would have a dose uniformity ratio of 1, where max and min dose is the same, but that's not going to happen. And so you have to go as close as possible to 1. And I show here examples what you have to consider. And these are only a couple of examples. There are a lot of questions that you have to answer, and the space is fairly big if you think about it. I mean, the first obvious question is, how do you orient your device in the beam? Is it along the longitudinal axis, or do you do it perpendicular? I mean, then, as I mentioned, you don't want to sterilize only one device. You want to have packages that are in the beam. How do you organize these packages? I mean, if you go along the longitudinal axis, I mean, you can put them all in a row, but then there are other possibilities. I mean, for example, in the case of syringes, I mean, it's a simple device, but is it better to go plunger, tip to plunger, tip to tip, or plunger to plunger? What is the best? And Then more questions is how close up can the devices be? Is it better to have them far away or very tight together? And these questions are also answered today. I mean, it's not new, but today this is mainly done by experience and measurement. And there's a lot to be said that there is a lot of experience out there, and the measurements work quite well, but there are some downsides. And first of all, to do measurements, you require a manufactured device, and you often also require more than one device. And as you know, in the development process, that is not necessarily easy to get, lots of devices. And then measurements are time consuming. You have to work with your contract sterilizer, they put you in the queue. And then if you want to change something, you have to start all over again. And then there's a possibility that changes are necessary. And as you all know, at that stage of a device development, changes are costly and delaying actually the time to market. So what Dose Insight wants to do is use modeling to answer these questions. And the obvious thing that is here, you don't require a manufactured device to do modeling. So all the questions that today pretty much require a device can be answered in silico. And also changes can be done before anything is built. So there's a upside to really go through this route. Just going a little bit into the details on how we do simulations. I mean, the first big question is, what is dose? I mean, dose is essentially the energy of electrons or photons that are absorbed in the medical device. So you have a high intensity beam of say electrons or photons, and these particles can interact with matter. They can be either absorbed, change direction, change energy, or generate new particles. And so you go basically it happens particle by particle. I mean, here energy is deposited, the particle moves on, and the energy that is deposited per mass is actually dose. So again, another event, and eventually the particle gets absorbed. So, and that goes for billions of particles to get the sterilization done. When we do modeling, we actually emulate this real process by simulating particle by particle and simulating how they get absorbed, and it's all by statistical nature. So first of all, we need to understand what our source produces. I mean, particles come out in a certain direction with a certain energy. We know all that from measurements. And then they enter the device, and we know the likelihoods that certain interactions happen, and we can model that, again, particle by particle. And this is a process that is known as Monte Carlo simulations. So it's a fairly well established in many different fields, and we introduce it here to radiation sterilization. Important is that we track each particle's history, we know where it's absorbed, and we know how much energy was deposited. And keep in mind, it's a statistical process. It takes a while before you really know what is going on in your dose map. And so you see how this is working. With that, I want to dive into our examples for virtual dose mapping. Here we looked at a simple medical device like a syringe, and we ask the question, which beam direction is better? Is it better to irradiate the long axis or perpendicular? And you see from this simple image that the dose uniformity ratio on the right is better than on the left. So probably going perpendicular is the better choice. But remember, I said it's not enough that you do it for one device. All devices need to be sterilized. And we can do that. We can simulate whole packages. I mean, here we chose a 4x3x3 package of devices and compare how it goes in the longitudinal direction or in the perpendicular direction, and find that it confirmed what we already suspected from the single device, that perpendicular direction is better. And this is only a short view of what can be done. Obviously, many things can be varied in these simulations, and they can be done fast and span a wide range of possible design parameters. That I want to close, and just summarizing that modeling can tell you a lot about your design process. You can enable its design for sterilization. You can compare modalities. You can figure out how you package your device, how you later on place dosimeter, which is also important. And in some cases, you may want to consider shielding of certain parts of your device. I think with that, I'm giving back to Daniel.
Great, thank you, Tobias, for that nice overview about virtual dose mapping and some of the benefits that it can offer you by incorporating it into your medical device development process. And with that, I'm excited to move over to the panel portion of the webinar. And we're very lucky today to have assembled representatives from all aspects of the medical device development process, from device to terminal sterilization. And so I'll give a brief introduction to them, and then I'll ask each panelist to introduce themselves. And I have sort of a prompting question about how you currently think about sterilization at your organization. So from the device side, we're very lucky to have Emily Craven from Boston Scientific. And Emily, with the way Boston Scientific currently does things, when do you start planning for sterilization validation? And how do you decide which modality is appropriate for a given device?
Yeah, thanks, Daniel. So as Daniel said, I work for Boston Scientific. I'm the director of global sterility assurance there. I've been there for about 4 years. And some of my opinions might be a little bit colored by the fact that prior to my time at Boston Scientific, I did work in the radiation, let's say, processing equipment design business. So I have experience with E-beam and X-ray and gamma, and even some dose modeling along the way. But to answer your more direct question, how do we decide, and when do we decide which sterilization modality is appropriate? I mean, as early as humanly possible is the answer, right? So at a company like Boston Scientific, we have, our life cycle processes for our products. And when possible, we try and actually get sterility assurance people involved right at the early design stages. Now, the reality of the situation is that sometimes we don't develop our own products, sometimes we buy products, or sometimes we have a legacy product and we're developing the next generation. So in those cases, we don't always have the same ability to sort of influence the design early on. But when we're brought in early, we can, sort of maybe push in a specific direction. Hey, why don't you look at this material versus that material? Or why don't you look at this packaging design as another one? Because what it can do is it can enable multiple sterilization options. And that's really from our company standpoint, that's what we want. We want to have as many choices as possible so that we can pick the best technology for that product.
Great, yeah, thanks for sharing that. Jumping to the end of the process, so terminal sterilization, we're lucky to have Larry Nichols here from Steritech. So Larry, if you could introduce yourself and then answer, with the customers you see coming to you, at what point in their product development process are they typically getting in contact? And at that point, have they really decided that E-beam is the way they want to go, or are they still open to considering other radiation modalities?
Hi, thank you, Daniel. I'm Larry Nichols with Steritech, the CEO, founder of Steritech, contract E-beam and X-ray sterilizer. Companies, when they come to us, a lot of them, they've already decided their product's going to market and they just need to use the sterilization method and haven't given it any thought at all to how they're going to do it. And it might be that E-beam is not the right choice. Maybe ETO is really the right choice, and we direct them that way. But if E-beam works, obviously we will accommodate them. Or if gamma or X-ray is a better choice, we'll push them in that direction as well. The problem is that they are not as advanced as Boston Scientific, for example. And many of the customers don't think about sterilization until the very end. And so that's why they are in this conundrum of how to do it and what are they going to do.
Yeah, great, thank you. From the sterile packaging design, we're lucky to have Sophie Strun here from QTS. And so, Sophie, if you could give an introduction to your role at your company, and then also, when you're designing packaging for a sterile device, how much do the specifics of the sterilization process play into that design?
Yeah, so again, I'm Sopak Strun from QTS. We do medical device assembly and packaging services, and I'm involved specifically in the sterility assurance realm of things, but I work quite a bit with packaging engineers at my company. And so, to echo much of what was previously said by Emily and Larry, it is very, very important to get us involved as early as possible. But of course, a lot of that doesn't happen for many reasons. It's so important, even just from the perspective of with sterilization efficiencies and costs, and making sure you maximize those in terms of your designs, because you have to balance things with making sure you can fit as much product as possible in order to be as efficient as possible in the process, but at the same time you have to balance that with sterilization compatibility, because when we're specifically talking about E-beam with the DURs or dose uniformity ratios, it's known that doesn't penetrate as well, and so you have to balance that, because if you try to fit too much product into, say, a shipper case, or if you don't use the right orientation, then that can lead to needing to process with a much wider dose range, and which could in turn mean much higher dose needed on the higher end, and that can cause issues sometimes with material compatibility. So yeah, again, get us involved as early as possible.
Yeah, that's interesting. And actually, because both you and Emily brought that up, it makes me think, what do you see as some of the reasons why some companies don't start thinking about sterilization earlier in the process? Everybody, just a question I thought of.
Yeah, it can be a lot of different reasons. A lot of times they just simply don't know. They could be a small company and they don't know to do that. It is something that we are trying to change as an industry, at least through the standards community that we've been involved in with the publication of AMI TIR, was it 100, I believe? Is that right, Amy, Emily?
Yep, TIR 100, yep.
Yep. So that's really trying to emphasize the need to get us involved in these early discussions, so that, while we don't necessarily design products for packaging within our roles in sterility assurance, we can provide inputs to ensure that those designs will work well and be efficient.
Another thing is, a lot of the device manufacturer or engineers, their goal in their mind is to design the device to meet the need that they're addressing. And sterilization is not one of those needs. And a lot of them will just use whatever sterilization method they used on the last project that they were designing for, which might not be the appropriate sterilization method.
Yeah, I agree, Larry. And so it's a combination of not only making sure you have the right people there that can make the decisions, but look at the motivations behind the decision. So you could end up with a company that has sterilization expertise, but their expertise is, yeah, we use that EO chamber down the street and that's what we use every time. And then all of a sudden you have a product that's not EO compatible anymore. Well, you're not asking the right questions at the right time.
Right. And I mean, what are some of the risks? I mean, the tagline here of this webinar is accelerating time to market. So what are some of the risks of not thinking about sterilization earlier in the development process?
Well, from my perspective, again, being a medical device manufacturer, you know, we make a lot of product. One of the risks is if you don't think about it, where are you actually going to sterilize it? Because one of the things we are running into in the industry is our capacity constraints. So if you haven't already figured out where you're going to sterilize it, and do you have a contract negotiated with that sterilization provider, or have you built enough internal capacity? That's my fear point with some of these things. And as a company that acquires other companies, that's the first question that I ask.
And we had a customer that came to us in August of 1 year saying they had this new contact lens. It's going to go to market in October and they want us to sterilize it. And we asked if they'd done any studies with radiation to make sure it wouldn't damage the product. And they said, no, why? Well, unfortunately, that was $10 million that went out the window because they did not get the product to market.
Wow. Yeah, that's a sobering experience, I guess. And yeah, so for the remainder of the panel, I think we'll just sort of have these sort of open discussions with everybody. And Tobias, who we've already been introduced to, will also bring perspective of virtual dose mapping. And I also see that there's a question in the Q&A. So I think we have a few prompt questions to discuss. And then at the end, we'll have a Q&A question. But if the audience thinks of anything as we have these discussions, please feel free to put it in the Q&A at the bottom of the Zoom meeting, and we'll make sure that we address those at the end. So that took a very scary turn, about $10 million and, you know, bankrupt companies. So what can we do about it? I mean, Tobias, maybe you can mention, like, what would virtual dose mapping offer to companies to avoid some of these risks about not thinking about sterilization early in the process?
Yeah, our goal is really to have that manufacturers have awareness that they can address it very early, and I think it can go along with the development process. As soon as you have your first CAD models of your device and think through, this is what we want to do. I mean, we would hope that people start just do a quick simulation. Does it make sense? I mean, one of the examples that I always like to say, I mean, there's maybe a metal post in the middle of your device, and you basically find through sterilization that actually gets a lot of attenuation, and it's probably not good. I mean, can you think at that point, hey, does it really have to be metal? Can it be some other material? And I think that probably is a very cheap change to do. I mean, not very much. You just change the material, and you're probably good with that. And later on, you don't really go through and redesign everything, because, as you know, it is extremely expensive to do that late in the process. And our model really is that we work with the developer all the way along until it comes time to really do the sterilization validation, which is done with physical devices. But our vision is that the models show, yeah, this is the DUR you get. You go to the sterilizer, say, please put the dosimeters on these locations, sterilize it, and then confirm that that is the DUR, and then you're ready for your FDA submission.
And that's an interesting one, actually. And maybe I can pose this in a controversial way, but could virtual dose mapping, so a simulation that predicts the dose distribution, could that ever replace experimental dose mapping?
I'm happy to tackle that one. So I don't like the word replace, I like the word supplement. And the reason I like supplement is that your model is only as good as your inputs and your assumptions, and you always are going to be in a situation where you need to test a model. One of the things with E-beam specifically is you can get really radical dose gradients. You can get very, unpredictability just because of the buildup that's happening and things like that. So again, in one of my previous lives before Boston Scientific, I worked on some dose mapping experimentation with coiled up tubing. And the way that the tubing was placed in the box, they were all on end. Every single dose map we did for that, we got different DURs, we got different results, because they really weren't true replicates, because just the slight difference in orientation of those tubes caused a completely different dose map. So I mean, maybe there's an answer there, because maybe, you know, with some sort of massive supercomputer arrays, you can do every conceivable orientation of product, right? But it can be very difficult to say, okay, here's a coiled up tube. Are the tubes all perfectly aligned? Are they off of each other by 5 degrees? How do you simulate that sort of thing? So there are some things that it's really hard to simulate. But I would say equally, there are things that are really hard to dose map. So I think that a combination of both of them is really what you need. And the way that I sort of approach, you know, virtual dose mapping is I use that to find my best orientation, my best packaging design, and I sort of define, okay, here's some minimum locations, here's some maximum locations. And then you put dosimeters there, but you don't have to put 500, not that that would even be practical. You put a number based on what you've predicted in order to validate your model.
Yeah, I agree 100%. Thank you, Emily, for voicing that. I don't well, I don't think it will replace it, but it would be great to use modeling to determine the proper orientation and then just do one confirmatory pass with dosimeters rather than doing the full 3 passes that is normally required.
And I just want to add, just, you know, based on my experience with dose mapping and E-beam, I mean, one of the barriers, well, one of the reasons why we're even talking about this today, specifically with E-beam and virtual dose mapping, is that one of the main barriers to why people don't use E-beam is the fact that it doesn't penetrate as well. And, you know, from a project-based perspective, it's much easier to just go down the gamma pathway because people know it works well. It's easy to get these products to market. But of course, now we're facing this situation with sterilization capacity in general, and you know, brings up the need to you have to look at alternative sterilization modalities and sterilization types, including E-beam. And so, you know, as an industry, we're forced to have to explore the use of E-beam. And so, you know, how do we overcome some of those hurdles to why E-beam hasn't historically been used as much? And, you know, in my experience, when we've had to use E-beam in the past, you know, before virtual dose mapping was even a thing, some of that exploratory or experimental dose mapping that you would perform up front to determine, you know, an optimal orientation of the product in the package or the package within the shipper case, doing all of that work can easily take months compared to, you know, the past, well, a typical gamma project. You can make broad assumptions for most products and not even have to worry about that. And so, you know, that's one of the biggest benefits here, you know, with if we can use virtual dose mapping and not have to spend months in determining what would be the optimal orientation and everything like that. And if it can take a matter of days, that's huge.
Yeah, thanks for sharing that. That's definitely one of the benefits we see. And to Emily's point, like to plan ahead in terms of make sure you have capacity or whatever beamline you want to use. You can do these types of simulations early on to really know, you know, I have to have it this way, it has to be, you know, this size conveyor, that kind of thing, to really make those decisions early on. So there's definitely some risk reduction capabilities there. Emily, you pointed out that some of the discrepancies you got between the simulated and measured dose maps was due to the fact that the tubing didn't match the model, the way that you had modeled it in simulation. What, in general, for a company like yours or like QTS or anybody who's considering using virtual dose mapping, what kind of data would you be looking for that would really make you trust the simulation?
Well, I would definitely look to take, well, ideally speaking, you might start with some of your existing products where you have existing dose maps. And then you'd go and you'd look and you say, okay, I put these 20 dosimeters in these locations. Will my model predict that same thing? That would be my absolute first step in terms of, do I trust what I'm seeing? And that doesn't necessarily have to happen with a product. You could, try and emulate or see what your OQ data. So if you're doing a phantom beneath the foam or something like that, you can look at those results against your model. That's the number one thing, right? Other things in terms of trusting it, part of it is, just the history of, if I take this one parameter and modify it a little bit, like, so energy, for example, like most systems that we're dealing with are 10 MeV. Well, is it exactly 10 MeV? Is it 10.2? Does it have a wide spectrum? Does it have a short spectrum? Modifying some of those and seeing what it actually does to your model, that might give you an indication of, hey, am I really far off here? Or is this a parameter that I really need to hone in on? Or is it something that I know is going to work? And so some of that figuring out how much detail you need in the model, part of it, I think, is really key as well. But really it comes down to, what can I predict? And then what can I measure with calibrated measurement systems to basically demonstrate that my model is giving me an output that I can then, basically calibrate to.
Yeah, thanks. And maybe Tobias, you can share sort of from those insights perspective, how have we, or what kind of data we've modeled to validate the simulations? Like, how do we know that we can trust what we're doing?
Yeah, I mean, that's excellent. And I mean, it's definitely an important part to have a validated model. And we are working on we have measured several phantoms and find in general very good agreement between measurement and simulation. And it turns out that probably the simpler the phantoms is, the better it is, because the more reliable you can really place your dosimeters on there and have less variation. And we also compared it to many products from clients, and seen also really good agreement, although we get exactly what Emily was mentioning, is that I mean, if you have a shipper that the dosimeter placement can be a little bit off, and then the agreement is less good. And I think it is probably both that the modeling is a little bit too rigid in assuming that everything is perfect, and then on the measurement side, that it's very difficult and labor intensive to really place these dosimeters exactly at the same spot every time.
Yeah, thank you for sharing that.
Yeah, one thing I also want to add is, there's often this fear of, well, does it represent real life dose mapping? Well, on the other side of things, with real life dose mapping, there's naturally going to be some variation from, you name it, dosimeter measurements, some of the other things that all of you have mentioned with where it's placed within the product. And, especially if you're talking about some of these more complex products where can you even fit a dosimeter in some of these tight locations, and you name it. And, in many of those situations, using computer models and virtual dose mapping probably would be more accurate in some of those types of scenarios. I mean, and that's why it's important. I don't view virtual dose mapping as something that would completely replace real life physical dose mapping. I mean, perhaps maybe one day when in the future, once as an industry we have a lot more experience, but at least at this point in time, I view them as things that would supplement each other.
I mean, maybe I add, we worked with a client that had a medical device that was packaged and consisted of multiple parts, and the multiple parts were not constrained completely. So just shake it and basically it had different configuration. And modeling can help with that too, because you can basically generate the different configurations. Hey, the part is here, now it's here, now it's here. Does it change the dose map? And we found that it was actually important, and the client actually opted to change packaging to make sure that all these different parts were fully constrained. So I think virtual dose mapping can address this variability quite well.
Yeah, it's actually an interesting point, because a lot of e-beam processing is double-sided. So if you have a process where something comes in and does a lovely little turn and then goes back, that's very different from where you take a box and you flip it over. You get different things happening in that package. And so you can have very wildly different dose mapping results if it's not constrained. So that's a really great example, Tobias.
Yeah.
So we talked a little bit about, what kind of data you'd need to trust the simulation, and the fact that it's also hard to trust the measurements because they're very challenging measurements. But say you had a virtual dose mapping tool and you were 100% confident that it was predictive. How would you change your current workflow? Like from, not just the packaging and device design perspective, but then also the sterilization validation perspective. Like Larry, for example, you said instead of doing 3 runs of the dose mapping, maybe you just do 1 and then use simulations for the rest. So in what ways would you change your workflow if you had this tool?
Well, it would be extremely helpful to us on many products. A lot of products that come to us, we can just look at it and know the proper orientation. But there have been a few jobs that we've done that one of them, it was actually 13 different passes before we finally figured out the correct orientation of the product. And that's a tremendous amount of wasted time and dosimeters. So to have the model already tell us which way to orient the product would be huge.
Yeah, for me, in terms of workflow, again, we don't always get in as early as we like, but we are looking at a lot of different optimization projects. And so we could use a tool like that to say, okay, given my box size or my packaging constraints, is there an e-beam system that maybe I could install myself that could be perfect for that device? Because that's another option. Pick the right energy for your device. Or if I'm going to say, all right, I'm going to use Larry's facility and it's a 10 MeV and it does this, what's the very best way for me to package that in order to optimize it? If I'm looking at multiple things at the same time and I'm given that constraint, because I'm not always given that constraint, and it's sometimes it's easy from the service provider standpoint to say, oh, just change your package or just change the material. But then when it's like 2 years later and however many tens, even more thousands of dollars worth of regulatory submissions and then machine and tooling changes, it's not quite as easy. So again, for me, it might not even be about the design for sterilization. It might be about optimizing the process, and that might be a way that I would use that tool.
And thank you. That's a great input, because the new system that we just installed has both 10 MeV and 5.8 and 7.5. And so we're experimenting with those different energies, which is finding better ways to process the product.
Yeah, that's a great example, because that's something that would be, again, very expensive to do experimentally, try those 3 different energies, for example. But with a virtual dose mapping tool, you can do it in a matter of, days, kind of thing. But we have quite a few questions from the audience, so I want to make sure that we have time to address some of those. One is interesting because we talked a little bit about what data would you need to trust the simulation. But there's a question from the regulatory perspective, like from the FDA, would you expect there to be any, or is there currently any validation that you need to do when using a simulation in a regulatory submission?
Not that I'm aware of it. And I think this is something that the ASTM group is going to be working on addressing after there is more data with modeling.
I know one thing that we're trying to do from an ISO level, 11137-1, which is the sort of governing radiation sterilization validation document. We're trying to change that language so that it speaks less about dosimeter or dosimeter system measurements and just more about dose measurements. So we're trying to be a little bit more flexible in terms of what actually a dose measurement is, and that can certainly open the door. But again, when we think about dose modeling, I mean, PQ, that's one portion of a very large submission package.
I mean, one aspect that is already allowed from a regulatory standard is determining where the minimum and maximum dose position is. So that can be done by modeling.
Yeah, that's in ISO 11137, and it doesn't put any constraints on the modeling system because ultimately you then have to put a dosimeter in that position to do the measurement. Another one, and this is maybe I can even address, but it talks about a question from the audience that's asking that in the medical physics, like radiation treatment planning industry, they've been using simulated dose for many years, and especially because it's something as critical as delivering dose to the human body. And so there's been a ton of validation work in that space. And so is that something that we can leverage in our industry in sterilization? And I think the answer is yes. And one of the ways that we're doing that is by using tools that are sort of the same fundamental tools, because ultimately we're simulating how radiation interacts with matter. And it doesn't matter if that matter is a medical device or a human body. And so, for example, at Dose Insight, we use the DN4 simulation engine. So this is, developed by a consortium of physicists across the world, and it's been used, to predict the existence of the Higgs boson, which won the Nobel Prize, but also for radiation treatment planning in medical space. So, using tools that are sort of battle hardened, tested over many decades, and developed by large communities. I think it's one of the ways that we as a sterilization industry can sort of lower the barrier to adopting virtual dose mapping tools. Another question from the audience is, what percentage how much does your choice of the sterilization technology add to the COGS of the device? Like what percentage of the COGS is related to sterilization, validation testing, and I guess terminal sterilization as well?
I think the bulk of it is actually the routine processing. Validation costs are a blip. And then it really depends on your device and the value of your device, right? Like if you think about sterilization costs as a percentage of a syringe, like what you're showing earlier versus like a pacemaker, it's going to be very different. But the thing that I would say, I mean, you can basically say anything between, 0% and 5%, right? I think that's usually what people are aiming for. But something that is notable is that transportation costs are often of the same magnitude as the sterilization costs. So again, it's relatively small in the grand scheme of things. And if the question is around differentiation between E-beam versus X-ray versus gamma versus EO, again, it's always going to depend on how you're processing, how efficiently you're processing. One might be better for one product and one might be better for another product from an actual costing standpoint.
Yeah, that's interesting. I didn't know that about the transportation. Do you factor that in when choosing which facility you're going to use? I mean, do you try to keep it local to where the manufacturing is happening? And
sometimes, I mean, the thing is that it doesn't always have to be local to manufacturing. It could be local to distribution, right? And so that's the thing. I think it's about, just figuring out what works for your supply chain. If you have to put it on a train or a boat or whatever anyway, it doesn't really matter if it's near the origin or near the destination.
Right, interesting.
And then of course capacity, who has the available capacity. So
yeah.
Yeah, and I mean that's something that's come up here a little bit about is the capacity gap, right? And so what do you guys see in terms of trends in the industry? And I mean, obviously there's talk of a big move to E-beam, but how big is that that shift from ETO or gamma to E-beam?
We're seeing a pretty major shift. And that's why we just opened a new facility and with plans to open a few more to try and meet the growing demand.
Yeah, there's definitely shifts going on. I guess I would just say don't underestimate the power of inertia. especially when you're dealing with products that are in gamma, because again, gamma to E-beam can be a difficult transition because of those different dose uniformity ratios and stuff like that. So if you have a choice between E-beam and X-ray, sometimes people go the X-ray route just because it's like, well, I know it's going to work. Because if it works in gamma, it's going to work in X-ray. That doesn't necessarily mean it's the best choice, though. And then certainly on the gas-based technologies, whether it's EO or otherwise, I mean, you're basically starting from scratch in terms of your design validation exercises and material testing, and that could be years. Like it could be, accelerated aging, it could be real-time aging to demonstrate that those materials are actually going to be appropriate for that radiation source. So.
Yeah, and there's quite a bit going on in the industry to promote the use of a wider variety of sterilization technologies. The FDA has been involved in a lot of those initiatives, standards groups, you name it. It's been a pretty hot topic.
Yeah, I mean, that's maybe also important to keep in mind. I mean, ETO came under scrutiny quite a bit over the last few years. I mean, there's this one lawsuit that was filed by somebody who lived in the neighborhood and got cancer and was traced back to the ETO facility. And she got awarded like $140 million or something.
300 million.
$340 million. And then there are hundreds of these cases pending. So ETO is under a lot of pressure right now. And similar, I mean, gamma has also some pressure because more about security concerns. That I mean, you have these big radioactive sources somewhere. I mean, that could be easily made into a dirty bomb. So the government has quite a bit of interest to curb these technologies a little bit.
Mm-hmm. Yeah, and all I would say is don't confuse bad press with a bad process, because EO and gamma and all of these technologies can be safely used and have been safely used, and there's always outliers, and I won't get into any legalities or anything like that. But the reality is that gamma is not going anywhere anytime soon, because it's a really, really, really good technology. And EO, there's a reason why it does 50% of the products out there, because it's a really, really, really good technology.
Yeah.
And what we want to make sure is that what we're reacting to is what's going to be best for patients, right? And what's best for patients is having access to safely sterilized medical devices that also consider the safety of the communities and the people that live there, and not banning things or turning things off.
Yeah.
Yeah, and we kind of have to do this as an industry to use more of alternative technologies to keep up with demand. The demand for medical devices with aging populations is only going to go up, and so the existing technologies, existing facilities, they're now going to be able to keep up with that demand.
We just have 2 or 3 minutes left. So if the audience has any more questions, please feel free to put them in the chat. There's another one here from the audience. Are radiation sterilization methods non-starters for devices that have integrated electronics?
I can say definitively no, because I've sterilized things that have electronics in them. So it's not a non-starter. It's just you have to have the right electronics, and you have to do the right testing.
Right. Yeah. Especially if they're passive electronics, there really aren't any issues whatsoever. It's the active electronics that can be sterilized under the right conditions for many of them.
I'm happy to hear you say that, because in our business, we obviously talk to a lot of medical device companies, and I feel like that's sort of a myth that goes around. People assume, can't use radiation. But with more and more devices having integrated electronics and them becoming more complicated, I think that could be a way to address the capacity gap as well, is just, allowing companies or educating companies that they can consider more modalities than they would expect.
And one of the solutions is to separate the electronics from the part that touches the body. And just have those pushed together, snapped together in the operating room. And the parts with electronics can be sterilized by another method.
Yeah, well said. Maybe in the last few minutes here, we can end with a question, which is, what advice would you give to a medical device OEM to stream or to make their lives easier when it comes to sterilization validation, you know, to reduce costs and risks. And what could they start thinking about now that maybe they're not already thinking about?
Test the materials as soon as you have an idea what materials you might want to use, and test them at different dose levels to find out at what dose level they break down, if they in fact do break down.
I agree. It's about early design choices, right? And there's great resources out there like AMETR 17, for example. They've put a lot of work into sort of updating with other sterilization modalities as well, you know, going beyond sort of the traditional radiation and EO as well, to Sopak's point. I mean, there's a lot of other technologies that are being developed. And what are those choices? What is that material choice that will give me the most options down the road?
Yeah, and then another gap related to this is just unfortunately, you know, due to confidentiality, you know, a lot of companies will guard a lot of this information. And so, you know, we always talk about this as an industry, you know, wherever possible, wherever your companies can allow, please publish data specifically on material compatibility, because that will help all of us as an industry if we can have access to a lot of that information. Otherwise, we're all stuck with having to do a lot of the same repeated testing on the same materials. And that ends up being, you know, kind of a waste of money.
And one final thing is, if you can afford it, validate your product to multiple sterilization methods, because you might want to start off using one, and if there's a capacity constraint someplace, you could switch to the other method.
Yeah, well said. And just to Sophie's point, there's Team NAVLO, which I think is out of PNNL, and they're sort of an industry consortium, an academic consortium about, and their goal is exactly that, to publish papers, academic papers, highlighting material compatibility with different types of sterilization modalities. So it's nice to see the industry working together to share that information. So I think we're at our time here. So just wanted to thank the audience for your participation in the webinar today, and for your interest in virtual dose mapping. And I'd especially like to thank the panelists here for sharing your expertise. I think it was a really interesting discussion, and I'm excited to hear sort of the same thing from multiple people across the spectrum of the product development process. And so I'm excited to see sort of what comes next for us as an industry. So if anybody in the audience is interested in finding out more about virtual dose mapping, please get in touch with Dose Insights at doseinsight.com or with Triple Ring, or feel free to reach out to us on LinkedIn, and we're happy to share some more information about that.
Thanks, Daniel.
Thanks, everyone. Amazing webinar. Everyone, we will be sending out the video, so don't worry. We'll send that out, just post. And one shout out, Emily. I thought you had a great mic drop on that ETO comment about not listening to the publicity, but the process. I was like, ooh, drop the mic on that one. That was awesome. So well done, everyone. And we look forward to seeing you all again very soon. Have a wonderful holiday. Bye-bye.
Thanks, Rachel. Bye.
Thank you, everyone.
Bye.